Hello and welcome to this Pharma On podcast, part of our DMPK Insights podcast series. I'm Barry Jones and I am the Chief Scientist for Global DMPK at Pharma On. With me today is Dr. Paul Morgan. Paul is currently Head of DMPK at Grünenthal and has previously held senior DMPK leadership positions at Pfizer, AstraZeneca and Heptares. Welcome, Paul.
Well, thank you, Barry, and thank you for the invitation to take part in this podcast.
So clearly we could have picked from a wide variety of DMPK areas that you've been involved in over your career, but I particularly wanted to focus on your work around drug attrition, and particularly as it relates to development and clinical phases of drug programs. So I'd like to focus on the two articles you published in Nature with other AstraZeneca colleagues. But firstly, before we start, can you tell me what is drug attrition and why is it so important to pharma companies?
Sure, let me do that, Barry. So drug attrition is simply the measure of candidate termination, i.e., stopping a candidate. It's usually classified by the phase of development, so preclinical development, phase 1, phase 2, phase 3. So the measurement of drug attrition usually and typically starts at candidate nomination, if you will, when the compound goes into preclinical development. And it's fair to say that this is an important measure of productivity for certainly for each company and pharma overall. And typically it's shared across the industry. All companies participate and provide their attrition information each year, and it's shared across the industry each year. So really, if it's used well, i.e., that information that companies glean, if it's used well, it can really help focus on improvement areas for drug survival within individual companies and across the industry. So that's drug attrition and drug attrition rates.
So, as I said, I do want to focus on the two articles you published from AstraZeneca, but it would be true to say that wasn't your first look at drug attrition. So perhaps you could tell me about how you first became an area that you were passionate about.
Yeah, so several years before the 5 hours work, while I was still at Pfizer, around 2010. I was involved in a team then that assessed the drug attrition rates at Pfizer. And what we were seeing was particularly high phase 2 attrition, with approximately, it was 70% of candidates were failing at that stage. And even worse, of those candidates that were failing, approximately 50% of them were we didn't understand and walked away from that phase 2 trial without actually understanding whether or not the mechanism of action had been tested. So it's the worst case scenario. Your compound has failed in phase 2. The company has invested a lot of money to that stage, and you don't know whether or not actually you've tested the mechanism. So our analysis identified, I guess, there were sort of 3 fundamental elements, if you will, that were fundamental to candidate success. And what I became passionate about, to answer your question, was it was sort of clear quite early on that actually DMPK and PK/PD could actually play a significant part in generating the necessary data and knowledge to help improve drug survival. So that was really motivating, not only to be involved in this important team at Pfizer at the time, looking into this for the company, but also as a DMPK scientist, seeing that actually there was a real profound impact that we could make to improving productivity. So we called these the 3 pillars of survival at Pfizer. That work was published in 2012. And for DMPK, the pillar that, if you will, was most relevant and most important, focused on generating exposure information at the target site of action over the desired period of time for the duration of effects. So very core the DMPK ethos and its impact. So yeah, so that's what really started me off in assessing, drug attrition, productivity, but also looking for ways in which DMPK could impact even more in this important area.
And I guess then, obviously, you moved from Pfizer to AstraZeneca. So if we then turn to the analysis you did at AstraZeneca that led to the first AZ publication. So what were the drivers that instigated this analysis at AZ? Were they similar to Pfizer, or were they different?
At the heart of the matter, it was a very similar, very similar driver, i.e., unacceptable drug attrition at AstraZeneca that had been occurring over about a five-year period. I think that was up until about sort of 2010. So yes, it wasn't. I sort of walked out of Pfizer, so to speak, into AstraZeneca. We had just or were just publishing the Pfizer paper. And then this work was started at AstraZeneca to look at the reasons for attrition over that period of time. But just at its core, it was that at AZ at the time, attrition was occurring at near or even above the industry norm at that time, and from candidate nomination to end of phase 3. So attrition, of course, as you will know, Barry, certainly at that time and still now, of course, it's an issue across all of the industry, but it was sort of a particular issue at AZ at that time. Phase 2 attrition was very high, and there were other areas as well. So that was the driver. In essence, we had a 5-year period where the productivity was if you will, that was unsustainable. Therefore, a comprehensive effort was needed to actually address what was going on here and how we could improve it.
So I mean, it was still very fresh in your mind from Pfizer when you went to AZ. So I guess, how do you approach an analysis of this type?
Yeah, no, absolutely. It's and what you're looking for is something that is comprehensive, that is objective, and is non-biased, and ultimately that it's set up to be able to lead you to some conclusions that actually you can take forward. Now, of course, this is being conducted, primarily by people who are in the company, who will have been involved in some of these projects. So I guess certainly some of the things that, and I should say that also, we were trying to cover somewhere in the region of 140 projects that actually had sort of, been conducted over that 5-year period. So there was a lot of projects to cover. So really, what we did was, and we got some input on this from from some external consultants, was to have a sort of structured questionnaire that actually could be used to interview people who had been part of those project teams, a structured questionnaire that could be used to uncover the sort of the relevant project data. And that was the way in which we aimed for it to be as objective and translatable as possible across all of the information. We even went as far in order to try to sort of avoid any bias of making sure that actually any senior leaders, for example, who had been involved in portfolio governance during that five-year period, that they weren't allowed to be on the team at all, so that actually it was as much as possible an independent and fresh pair of eyes that was doing the analysis. So yeah, so that's what we did. That took quite a number of months to conduct the interviews, to get all of the information, and then we started to sort of pull it together to see whether or not we could identify any trends, and ideally, would we be able to identify any determining factors by which productivity could be improved. So that was the analysis.
So I'm pleased you picked up on the bias piece, because that interests me. Because I guess, having been part of a number of drug discovery projects over the years, you do become very passionate about your project and wanting your project to succeed. So I guess, there's trying to get an objective view, after action review, if you like, of a project could be quite tricky, to pull out the key strands.
Absolutely, and of course, everyone who's ever been involved on a project, you and I have many times. Many of the people listening to the podcast will be the same. Every project is pushed forward with a chance. Yeah, the people on the project firmly believe that there is there's a good chance. And there are many reasons why to be thinking positively about it. I guess the challenge they're in, and of course we've seen it, and certainly what I guess we were trying to address within this is that more often than not, human nature, and particularly when you're on a project, is that you can tend to always think about the positive end of the range. You will think, okay, well, if only this we get this positive data, or we get that positive data, then we'll be in a great position. And but as we know, of course, you can get a variety of outcomes on the project, and sort of that's what we were trying to address. Well, and it's as much about actually trying to find, as I say, the reasons were and the ways in which you can improve the probability that you will be successful by having particular types of data. Particular milestones would increase the probability, and maybe before this, certainly in the scenarios that I've been worked in, that wasn't necessarily always the case. Projects were maybe hoping that certain types of data would come out that would be very positive, of course, but of course in a lot of cases they didn't get data like that. They got worse data, and then obviously a lot of the cases had to stop the projects.
No, I have been there. Okay, so having conducted this analysis, so what were the key findings?
So I guess the top level finding was that the overall attrition rate from preclinical development to end of phase 3 was very high, or to say that another way, productivity was low. So over that period of time, there was only around about 2% of the projects that had started that actually ended up reaching the end of phase 3. Now that's clearly very low productivity. Now, relative to the industry norm, it was lower, but the industry norm at that time was only about 4 to 5%. So so the industry had for sure had an issue in terms of really it was less than 1 in 10 projects were actually sort of surviving to the end of phase 3. But at AZ, we were even on the lower end of that industry norm. So that was the top level issue. Digging deeper into the analysis that we did, you could clearly see that there, at particular stages of development, there were very clear issues. So, for example, safety-related attrition was the key reason why projects, the majority of projects, had failed both in preclinical development and in phase 1. So that was clearly at that stage of development, something that needed to be looked at. Of course, in some ways, safety-related attrition, certainly in preclinical development, more than likely is going to be the major reason why projects phase at that stage, because GLP toxicology is the key study that tends to be done between candidate nomination and first in human. But what we were finding was that in a lot of these cases, these were safety signals that were being uncovered that the team hadn't necessarily thought about or hadn't necessarily seen before. So it was unknown or unobserved safety. So that was one particular issue. Lack of efficacy in phase two again was a sort of really clear a clear issue as well, and probably a significant high part of the attrition problem was in phase two. So yes, so those were the top level issues with regards to where the attrition was occurring. What the analysis then showed when we compared projects that had failed versus those projects that had progressed at each of the stages, there were some really interesting trends that actually came out of that and that were apparent. For example, for those projects that failed versus those that survived or progressed, confidence in the target was lower. The human genetic linkage of the target was lower. The availability of biomarkers, both for preclinical and clinical investigation, was lower. Safety signals became apparent too late. And overall, the PK/PD understanding was just not well established. So it was a real eye opener for us in terms of actually just those systematic gaps in the data and the knowledge, even for projects that were entering late stage development. So identifying those trends actually showed two things. One showed that actually the analysis and the way in which I described the analysis and review had done what we were hoping it would do, that identify some trends, and secondly, that really then led us to, I guess the 5 Rs being born, so to speak, because you could go through each of those trends and sort of say, okay, this is what we need to focus on. We need to focus on the right target, the right tissue, right safety, right patient, right commercial, with the expectation that the data and knowledge on each of these 5 Rs would be provided going forward by all the projects for every candidate, and they would continue to be developed and updated through the development life cycle. So that was the start, if you will, of the next phase in terms of implementing the 5 Rs. But that's what came out of that first analysis.
Okay, so we've got 5 Rs. So are all the 5 Rs born equal? Or are there some elements that the analysis showed were more important than others?
Yeah, no, it's a really good question, and I've been asked that sort of quite a number of times. And I mean, the intention was that each of the R's would be worked on at the relative stages of development. So it would probably be, in answer to your question, it would be fair to say that for example, in the discovery and preclinical development phase, then for sure, right target, right tissue, and right safety were the most important R's at that stage, and therefore they received the most focus. That's not to say that you didn't need to be thinking ahead in terms of the patient, in terms of commercial, the target product profile and so forth needed to be thought on. But those were the most important, at that stage. And then just sort of, I guess, thinking a little bit about DMPK being parochial for a minute. The right tissue, R, if you will, was actually the one for which DMPK information and knowledge was incorporated within that. And what was really interesting about that was, as we worked through it, was that actually that right tissue R would be needed to be focused on all of the way through the development cycle, preclinical and clinical. You would need the sort of development information around DMPK and PK/PD. And again, as a DMPK scientist, while I was obviously thinking about it for the overall company and all of the 5 Rs, that was really neat to sort of see again, here's a way in which DMPK sciences could have an impact through the whole development cycle. So we published that in 2014. Obviously, by that stage within the company, the implementation had got underway. But as I say, that paper is what we ended up publishing in Nature Reviews Drug Discovery in 2014.
Okay, that's great. And So before we move on to the second publication, are there any other important elements from the first paper that we've not covered?
Yeah. What I would say to that is that I mean, clearly, as I described earlier, what this identified were there were data gaps. There were data gaps and knowledge gaps at various phases of development around the projects. But from the outset, we were sort of keen that shouldn't just mean to the projects that they do necessarily, more and more studies earlier and earlier. It was also trying to instill an ethos, a mindset where actually we would sort of, work smarter, if you will, not necessarily do more. Of course, there was an element of where you had to front load, some packages of work that previously had been done later. So there were some instances where you were front loading, so particularly, I guess, around the sort of the safety related attrition and front loading some of those studies. But the work smarter bit. So really, what we were trying to foster was a mindset of let's now be using this as an opportunity to use new technologies that are coming in. So we saw a real upsurge in using advanced imaging, for example, in terms of trying to sort of identify information around exposure and target engagement and biomarkers, using advanced human in vitro models where we could. And that progressed all of the way through it as those advanced, human in vitro models matured. And I would also say there was a real upswing at this stage in the incorporation of sort of PK/PD, quantitative systems pharmacology. There was a real investment in that actually as a result of this, so that we could again, hopefully work smarter and more quantitatively as we implemented the R's. Yeah.
Thank you. It would be remiss of me at this point not to remind listeners to the podcast that as part of our Pharma On webinar series, we have a webinar from Neil Benson and Simon Taylor on PK/PD modeling. So if people want to find out more, please go to our Pharma On webinar series. Okay, so moving on, and thinking now about the 3 pillars and the 5 R's together. So what do you see as the kind of the common strands between the 2 analyses? And actually, where do you see the differences?
Well, in terms of the commonality and common strands, actually the clearest synergy was around those elements related to DMPK and PK/PD. Both the 3 pillars and the right tissue from the 5 R's were concerned about demonstrating exposure at the target site of action, and it being really important to generate that relevant exposure data, i.e., free exposure at the relevant tissue, and not just assume, as a lot of projects had done based upon plasma exposure, that actually they would know what the relevant exposure over time was in the target tissue. So that was a clear synergy between both. Hopefully, not a clear synergy just because I was on both project teams, but because it was it was clear that was a fundamental understanding that you needed to have around the relevant exposure at the target tissue over the duration of time that mattered for effects. And then having that data then to demonstrate that you were getting target engagement and functional pharmacological activity, which was commensurate with that target exposure. So I'm sure a lot of people that would be listening to this and sort of say, well, yes, of course, that's obvious, and it is, and it's fundamental. But certainly at the time, and I would imagine, even still, there are challenges, of course, sometimes in generating that data, particularly clinically. Yeah, when we're talking about generating, data at the target tissue, of course, in clinical studies, that's not necessarily going to be feasible. So you have to have a real good understanding of that from your preclinical data and your PK/PD model, and the PD biomarkers that you're going to look at in the clinic, in order to be able to sort of, for that all to hang together in an understandable way. So I would say, so that was a clear synergy and that was a clear commonality. I don't think that there were necessarily any differences. To be fair, the initial Pfizer and assessment we did was very much focused around sort of phase 2, because that's where there was a real issue at the time within the company. So there were only 3 hours, whereas I guess the AZ one was focused on the whole sort of project life cycle. Subsequently, of course, other folk more recently at Pfizer have actually expanded on the 3 pillars and published further papers on that. So I would say, maybe not surprisingly, there's more commonality than there are differences, because these are issues that we're sort of facing across the whole of industry.
And so, just listening to what you were saying there, so some things like the free drug theory are enshrined within both the 3 pillars and 5 Rs as fundamental tenets that support successful drug discovery and development.
Yes, very much so. Of course, it is appropriate to sort of say that certainly at the time, the majority of the assessment here was on sort of small molecule projects, and when we're talking about the sort of the free drug hypothesis and the type of data here, then more often than not, that's what we had in mind. It was for small molecules, but absolutely, I mean that was a clear outcome from this, of where there were sort of projects that hadn't taken that on board. Back to what I'd said about, which was part of the analysis of both, was not only where projects had sort of, if you will, failed at phase 2, but also had not tested the mechanism. In quite a number of those cases, it was because that actually there was a, if you will, an incorrect assessment on the concentrations that would be needed to actually for efficacy, i.e. total drug versus free drug. There were other factors there as well in terms of not being able to get up to the appropriate dose, but very much, free drug, in the target, as I say, in the target tissue over the appropriate duration of time was a core tenet of this.
Okay, so now we turn the clock forward to 2018, when the second AZ Nature publication came out. So the 5 Rs had been in place for around 7 years by the time of this publication. So is this essentially a review of progress and impact?
In many ways, it was, and we were really keen to, obviously internally within the company, to evaluate and demonstrate the impact of the 5 Rs in terms of improving overall productivity during that sort of period. And while we were doing that, as we went along, this actually was a chance to actually pull all of that information together in a really sort of comprehensive way. But equally, we felt that there was a need, almost an obligation, if you will, to be transparent to the external community around the 5 Rs, to actually see whether or not this was this was as impactful as we hoped that it would be. So we were really keen to publish again, to publish in Nature Reviews Drug Discovery again, because again, it being a high impact journal, then that would sort of garner the most readership, if you will. I mean, fair to say it was a challenge, because at this stage now, actually, we're talking about not just a new set of metrics around productivity, but you needed to demonstrate that with real world examples that you were able to publish. And of course, sometimes within industry, that's not always easy. And obviously, we had to sort of be open to publishing both positive examples, but also where there may have been sort of, failures as well. So it took quite a bit of time to sort of pull that paper together in a way that actually it could be fully accepted. So, but yes, in essence, that's what it was. It was looking back to see how successful it had been, and what were the further lessons we could draw from that.
Okay. So, and I think, that speaks volumes that AstraZeneca was willing to publish successes and failures to emphasize this. And so what were the major findings on the impact of the 5Rs?
Well, the good news was that overall there was significant improvement in productivity, as I say, from that preclinical development to end of phase 3. That overall productivity had improved to 19%. That was what the data sort of showed us. So from around about 2% to 19%. And I should have said that the period of assessment for the second paper was 2012 to 2016. So it was a four-year period. I think that covered around about 100 projects. So it was still a good number of projects from which you could sort of draw some clear conclusions. So overall improvement in the productivity, which was great to see. And then within this, we were also able to sort of see that there were sort of clear improvements in all of those categories, which actually had been sort of major issues during the first phase. Safety-related attrition in preclinical development in phase 1 was reduced significantly. Phase 2 attrition was significantly reduced. So yes, so all of those, there was a clear positive upturn in the success and the survival, with attrition being reduced. And then just maybe a few words, I guess, about DMPK related productivity. Over that period of time, it was really pleasing to see that actually there was an improvement in the accuracy of human PK predictions. The methodology that we were across the industry, of course, improving all the time, was leading to an improvement in the human PK predictions, and that was sort of great to see. And then, furthermore, I guess, I've mentioned this a few times, the sort of the PKP understanding was also improving all the time, i.e. the relationship between, drug exposure, free drug exposure at the target site, as we've discussed, and the expected downstream pharmacological effect. That was a real focus for all of the projects during that time. And ultimately, in the clinic, where you were looking for that to actually play out, was whether or not you were able to demonstrate proof of mechanism in early clinical development. And one of the most pleasing aspects of this was that that's what we sort of saw. So during that period of time, we had sort of just short of, I think, 30 positive proof of mechanisms in that second phase, I think, compared to just around about 5 in the first phase. So we were seeing a real improvement in the clinical phase, which of course meant that all of those projects with the positive proof of mechanism were able to progress into the next stage, into phase 2, into phase 3, with much greater confidence in the doses, with greater confidence that they would have sufficient safety margins and so forth. So yeah, those, I would say, were certainly for me were some of the key learnings and the key observations that we saw within the second stage, which of course was very positive.
Yeah, I mean, an increase in productivity of nearly 20% is quite spectacular. So this second analysis, or I guess this review, did it lead to any revisions to the existing Rs or the development of any new ones?
No revisions, I would say, per se in terms of the technical aspect of the the 5 Rs beyond I guess the expectation that you would have an evolution of those, certainly an evolution of the technology that was needed in applying those 5 Rs. And also, obviously, as time went on, we were then applying them to a broader range of modalities. Yeah, as I said, starting with small molecules, but then, of course, we would have oligonucleotides coming into that, RNNi, monoclonal antibodies. So not necessarily new Rs, but applying them, if you will, in a different way with new technologies. But actually, what I would say is, and maybe this is sort of back to the point you were making, and a lot of people made at the time when we were presenting this. Wow, such an improvement in the productivity. That came about just by generating and focusing more on those data. And I would say that was one element of it, but a really significant element of it was that actually the right culture needed to be in place. You could almost say that was the 6 star. And that started from the top. And I would say sort of kudos to the senior leaders in AstraZeneca at the time. They set the tone by saying that actually projects had to have data around the 5 Rs every time they came to a portfolio governance meeting. And if they didn't have that data, then they were more than likely not going to get the support to continue. So that set the tone right from the start that this was there and this mattered, and project teams then absorbed that and took that on board. And made it their business to actually then be sure that they were generating the data around the 5 Rs for each of the phases of development. And then functional groups like DMPK embraced it by making sure that actually they were advancing their technologies in order to be able to actually provide that type of support. So I would say that yeah, ultimately you could say yes, there was a new one, and it was the right culture, and that was so important as I look back on this. So important that support and that role modeling was there from the very top of the organization.
So kind of like a revolutionary or evolutionary change in culture from top to bottom of the organization.
Yes, yeah, that's what it took. There were bright scientists in many, many different sort of companies. So it wasn't necessarily that, and of course, all of the same technology you say is available to us all. It's the way in which you apply it. And it's the focus and determination you sort of bring to it. And as I say, that culture, that ethos that was there, certainly during the period of time that I was AZ, was clear, and certainly probably one of the most pleasing times, I would say, of my career, to actually to be a part of that and to be a part of the part of it within the company and actually implementing these and deploying as I say, this data and knowledge within the framework of the 5 Rs was really exciting and really rewarding.
So you've mentioned DMPK and DMPK's role in both analyses and indeed in productivity going forward. So I've got now just 2 or 3 questions around DMPK. So has DMPK impact evolved in contribution to improving R&D productivity?
I firmly believe it has. Certainly in my experience, of course, as you know, you and I have been around for a while, shall we say? So DMPK has always, of course, certainly in our time, at Pfizer and AZ and other companies, we've always been sort of, very much at the heart of particularly drug discovery teams and project teams. But I would say that impacts. Where I see the impact significantly evolving for DMPK is in the relevance of the human PK, PK/PD, and dose predictions that typically are actually for which DMPK scientists have a core accountability. We're not the only ones that contribute to that, of course, in terms of the data that comes into it, but typically that accountability lies with the DMPK organization in terms of putting all of that together in a quantitative way. And that's through the whole project life cycle, as I said before, from preclinical through to clinical. And so the best performing DMPK organizations, I would say, are those that actually have a clear continuum of that PK and PKPD understanding and the modeling, with forward translation, back translation, from discovery to clinical and back and forward. That's where I would sort of see as the best impact of DMPK, when that accountability is taken to heart, and for which, as I say, it's clearly, even though discovery and preclinical and clinical may sit in different parts of the organization, actually they're working as one to provide that impact.
Hmm. So what expertise of DMPK needed to evolve?
I would say that the area where maybe DMPK had to evolve the most, and now thinking about certainly with the 3 pillars and then the 5 Rs, in order for us to be a really significant player in that PKPD understanding and implementation for efficacy and safety, was actually in the biology and pharmacology. DMPK has always been closely aligned with chemistry in terms of obviously, choosing the compound and optimizing the compound, and of course that is still key, for sure. But certainly my observation was that the DMPK scientists, certainly those at the project interface, those involved in, certainly in PKPD assessment and understanding, really needed to have a sort of deep understanding of the biology, much more than maybe we ever used to have, in order for us actually to be a key player in that way. So I would say that now that's probably I guess an expectation that we would be as equally well versed and able to converse with our chemistry colleagues as well as our pharmacology and safety colleagues, and have a sort of real deep understanding of the target related biology and off target as well. So that's where I've probably seen the biggest evolution.
Okay. And in terms of technology advances, what have DMPK needed to employ to impact?
Yeah, I mean, just calling out, I suppose, what I have seen and what I've maybe been most involved with in terms of implementation of the 5 Rs. I mean, I think I'd mentioned this earlier a little bit, is that using, because often when you're talking about actually trying to determine target exposure, that it isn't necessarily enough just to, for example, in a preclinical study, take tissue homogenates and generate concentrations of the whole tissue. That is in some cases isn't just enough, and so the deployment of advanced imaging technology, mass spec imaging, other types of imaging, I've seen a real surge in that, and that being employed in a very, very useful way, giving us actually information around the sort of the distribution of the drug over the target tissue, which isn't necessarily homogeneous, and where that aligns with actually, PD effect and so forth. So that's been a clear improvement area, I would say, or where we've had to evolve as DMPK. For sure, back to the modeling bit, acceptance and deployment of more sophisticated PBPK, PKPD modeling, quantitative systems pharmacology has been core to this as well. And maybe last but not least, something that maybe doesn't get talked about enough, perhaps, is the bioanalytical capability. That's core and fundamental to what we do within DMPK. We're a quantitative sciences in measurement of drug concentrations, etc. And bioanalysis had to evolve significantly, I would say, to cover both PK and PD quantitation across a range of modalities. Has sort of saw, certainly in my experience, a real uptick in that.
Okay, thanks for that, Paul, and many thanks for your time today. This has been a fascinating and insightful discussion.
Well, thanks again for inviting me, Barry. You're really happy to contribute, and thanks to Pharmon actually for I should say on behalf of myself and maybe more grandly DMPK scientists. Really nice that you have this series of podcasts and webinars. I know they're well appreciated within the DMPK community. So thanks for that.
Thank you very much. So thank you all for listening to this episode of Pharmaron's DMPK Insights series. We would like to remind you that our webinar series is also available on demand, covering a variety of key questions related to DMPK science in drug discovery and development. Stay tuned for more podcasts in our Pharmaron DMPK Insights series. Thank you and goodbye for now.